synthesis. Yes, this came a long way. Since grade 8, we have studied the cell and the nucleus of the cell with its important genetic information. Now we know that the genetic information is made of DNA. We also know that DNA contains a specific code. This code is in the sequence of the bases, the nitrogenous bases. And today we are going to see how that code on the DNA is used to make proteins. Protein synthesis comes with the expression of the gene. The sequence of the nitrogen bases will eventually tell us the code of the protein. I would love us to quickly look at proteins. Yes, we know that proteins are made up of a specific sequence of amino acids bound by peptide bonds. This sequence will then be folded into the secondary structure of a protein, which is held together by hydrogen bonds. Then we have the third or the tertiary structure, which make these bonds into a three-dimensional structure. Now this three-dimensional structure is held together by disulfide bonds and also hydrogen bonds. That is the tertiary structure. But proteins also have a quaternary structure. We have different polypeptides making proteins bond together. We need proteins because it makes up living material. We know blood contains hemoglobin. The brain needs the channel proteins in order to get the brain signaling small molecules to enter the nerve cells. We know at the synapses, this channels is important for calcium and sodium ions to enter. Enzymes need to digest inside the mouth, inside the stomach, the small intestine. But enzymes will be all over in the mitochondria. Enzymes will catalyze reactions, all made of proteins. Then we have clusters of proteins to help the cell copying genes and making cell division and new protein synthesis possible. Antibodies, proteins that helps with the immune system. We call them immunoglobulins. Cell messengers, the receptors at the cell surface membrane that needs to communicate with the inside of the cell. And also hormones are messengers, chemical messengers made out of proteins. Then we have the muscles and the hair and nails. We also divide proteins into globular and fibrous proteins. And just to help us to remember, let's add hemoglobin, globular protein. So it's a spherical shape. The channel proteins, globular. Enzymes, globular protein. Clusters, globular protein. Antibodies or immunoglobulins, globular proteins. The hormones and the receptor proteins outside the cell membrane, globular proteins. And then we have the fibrous proteins, the keratin in the hair and nails, and the hooves, feathers of other organisms other animals. And then the muscle proteins are also fibrous proteins. Wow! Now we did a lot of protein revision. That is the recipe, dear grade 12s. Repeat, repeat, repeat. Now, we are going to see how these proteins can be synthesized. So, we have a sequence of amino acids to be connected by a peptide bond to one another, and eventually it forms a polypeptide. The polypeptide is folded and bonded together to make a protein molecule. We have 20 different amino acids, and how do we get the amino acids? We get the amino acids by what we eat, right? So, this amino acids will be part of the ribosomes. The ribosomes will take all these different amino acids that we got from what we have eaten, the proteins that we have been eaten, will be digested, and I will have lots of amino acids in my blood. Now the ribosomes will have the job to assemble them into the correct proteins. Where do I find ribosomes? On the rough endoplasmic reticulum, and also lying free in the cytoplasm. If now I am looking at the functional components of protein synthesis, we of course will start with DNA, the double helix with a specific sequence of nitrogenous bases. This nitrogenous bases for the DNA, cytosine, that will always bond with guanine, and adenine, that will bond with thymine. Always bonded like that. It is not a random sequence, but this sequence will hold the key. This will be the code for a specific amino acid. 3 of them. Okay. Then we know that we have RNA, and especially the mRNA needs to link with DNA at a specific stage. So, it also have cytosine, which will link with guanine when it needs to link with the DNA. Adenine will now link to uracil. Then we have the RNA. So here we have our DNA molecule. DNA contains cytosine and guanine. Guanine and cytosine are linked in this double helix. We call them complementary strings. They do not look the same. On the one side, if I have cytosine, on the other hand, I will not have cytosine but guanine. Then I have adenine and thymine, which will link together. Okay, then I have an RNA strand, which is a single strand. And here I see, okay, cytosine and guanine resembles DNA, adenine resembles DNA, but uracil is a different nitrogenous base or nucleobase that I find on RNA. So why? To make protein, we need the following to take place. We need transcription. So, transcription means to copy the genetic information from DNA to RNA. We call that transcription. RNA will copy the genetic information, but the copy on the RNA will be the complement of the DNA message. You agree? That we call transcription. Why do we have transcription from DNA? Because DNA contains the genetic code for protein. It is needed to get that message to the ribosomes that is outside the nucleus. But the DNA is large and the double helix cannot leave the nucleus. So therefore, what needs to happen is the DNA needs to unwind, unzip, and to unzip it needs breaking the hydrogen bonds between the nitrogen bases. Now the messenger DNA will come and make a copy. Again, it is not an exact copy, but it will form the complement. It will form the complement of the message. Because the single strand of RNA can leave the nucleus through the nuclear pores. So what will happen is, if we can just go back to our previous slide, the guanine of RNA will link to cytosine. So let's make a note here and to help us to remember. The guanine on the RNA will link to the cytosine on the DNA. Right? There we have RNA. Guanine will copy the cytosine. The adenine of the RNA will link to the thymine of the DNA. The uracil on the RNA will link to the adenine. Right. The cytosine on the RNA will link to the guanine on the DNA. I hope you do understand that. So now we have a transcription, a copy of the DNA that opened to have the part with the specific code for a specific protein open. So, transcription, a part of the DNA unzips, unwind, unzip, breaking the hydrogen bonds. mRNA comes and take only the part with the code on and copy the code from the DNA, but in a complementary message. It will copy the complement and there will be no thymine but uracil. That is what we call transcription. How does it take place? There are free RNA nucleotides inside the nucleus, and that was produced by the nucleolus. Made the RNA, the free copies attach in the correct complementary order, and now it is called transcription, leaving the nucleus. The mRNA goes through the pores with the message. It's taking this message to the ribosomes. In the ribosomes, there are transfer RNA. Transfer RNA. Which is, this is a tRNA or a transfer RNA. We see that attached to each transfer RNA is an amino acid. So this amino acid will only be attached to this specific 3 bases. This 3 bases is called the anticodon. This anticodon inside the ribosome will fit 3 specific sequence of a mRNA, and that we call a codon. On the RNA, mRNA, we have a codon. And just to help us to remember that it is 333, they call the 3 there that will belongs together to form a codon to fit a specific anticodon. On the DNA, they say there must be a triplet of codes on the DNA that will form the codon of the mRNA. On the codon of the mRNA, there will be an anticodon. Attached to that anticodon, we have the specific amino acid. And now we call this translation. When the codon is fitted out with an anticodon, now there will be a specific order of amino acids linked together, forming a peptide bond, and that is translation. So, let's quickly talk about translation again. The decoding of the mRNA, 3 bases of mRNA, will attach to a tRNA. The tRNA is like hooking 3 specific nitrogenous bases, because this 3 had that amino acid attached to it. As this one is leaving, a next one can attach in the ribosome. And there I will then have a specific sequence of amino acids which will form a polypeptide string. That is translation. The mRNA coding for a specific tRNA with its attached amino acid that will be sequenced inside the ribosome. That will be called translation. The transfer tRNA carries amino acid from the cytoplasm to the ribosome. It will attach to a specific 3. These specific 3 will be called a codon of mRNA attached to an anticodon on the tRNA. So where did this amino acids came from? That is now in the cytoplasm. That came from the proteins that I ate. It got digested or hydrolyzed inside my digestive system, absorbed by the blood, transported to my cells, and now the amino acids can get into the cells. This tRNA, together with the code on the mRNA, rearrange all the amino acid so that I can have a new protein with a specific function in the human or any other animal or plant cell. And again, a series of 3 adjacent bases on the mRNA is a codon, and a series of 3 nucleotides that are complementary to the codon will be on the tRNA. What will be the complement of this codon? G will have a C, C will have a G, A will have a U, because we are talking, and this will all be attaching to a specific amino acid on the tRNA. Right. So each tRNA codes for a different amino acid. Messenger RNA do transcription. mRNA is transcribed in the nucleus. The mRNA can leave the nucleus through the nucleopores because it is small enough. It moves through the cytoplasm and join the ribosomes. Inside the ribosomes, which is the factory of proteins, the codon of the mRNA will be translated by tRNA. The mRNA enters the cytoplasm attached to a ribosome. Translation begins at AUG, which is a start codon. Each transfer RNA, tRNA, has an anticodon whose bases are complementary to a codon onto the mRNA strand. The ribosome positions the start codon to attract its anticodon, which is part of the tRNA, which binds methionine. The ribosome also binds to the next codon and its anticodon. And then I have an assembly line of amino acids joined by a peptide bond to form a polypeptide. When I have a polypeptide and it completes its 3-dimensional structure, I have a protein. So we can use a codon chart, which is fortunately set by all the clever scientists that researched protein synthesis, and now we can discover which amino acids will fit. So when I have CAC, I look for C in the first base, A, and here I have a C, right? Third base. So it will be histidine. You agree? You can pause the video and see if you can find another one yourself. C. Again, C. The second one is C and an A. C, 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 A. There it is. So it will be proline. U. Let me use another color so that we do not get confused. U, G, U, G, A, U, G, A. UGG, first UGG. UGG is tryptophan, and then I have a stop in UGA. So, you got that? Right, then the next one is a different type of chart to help us, and we start in the middle. A-U-G. Aha. So the first one will be methionine. Again, you can try and find them on your own, and then play the video again to see. There we see methionine, asparagine, aspartic acid, and then a stop. To capture this, we have DNA needs transcription by the mRNA to get the code of the gene to the mRNA. mRNA will be translated by tRNA, and that will form the protein. Add your own sounds. RNA polymerase doing the unzipping. RNA subunits produced by the nucleolus. So I added a quick revision sheet for you so that you can quickly see and bind the correct names in your pathways in your brain, dear class. Please print this one as well as the added summary that also will help you to quickly revise it and the worksheet attached. Please try your utmost best. I think that you then repeated it enough, and if there are any more things that is not clear to you, just watch it again or read through your textbook.
ShopRite has always been here for you, and our promise to bring you low prices on what you need most will never change. Get an essential food hamper that includes Sugar King white sugar, Top Score instant porridge, Top Score maize meal, Rice King parboiled rice, Bakery brown or white bread, Right Brand sunflower oil, Clover Numel dairy blend, Polana spaghetti, Romi low-fat spread, and Imana regular soup. All 10 for just $110. only at ShopRite. One stop, one safer shop.
We always love to hear from you. Send us your views, comments and videos with your name and town to the One Africa TV's WhatsApp on 081 200 6659 or send an SMS to 555. One Africa TV, it just gets better.
Good day. I hope you are busy and that you really put an effort into this work. First, I would love us to get the answers to the diagram that I ended the previous video off with. So if you can remember all the different diagram labels, then you will be able to start to understand what is happening. Grade 12s, so there you will have the label DNA. There we have DNA. And DNA will be transcribed, or transcription is the process where the DNA unzip due to RNA polymerase, and I will have copy of the complement made by the mRNA. Remember that it is loose nucleotides of our mRNA that will attach. Now the mRNA can leave the nucleus due to the fact that it is only one single strand, and it will be transported to the cytoplasm. Now the mRNA will reach the ribosome, and 3 bases of an mRNA forms a codon. Now we will have the process of translation. The process of translation will take place when the tRNA, with its specific 3 nucleobases or nitrogenous bases, will attach to the anticodon. Attached to this, there will be a specific amino acid, and as this amino acids are in a specific sequence linked via the tRNA, we will eventually have a protein. I hope that you got this one, and while you are doing this, repeat. Read in your textbook to find the correct answer. Now I want us to look at our worksheet. We did a worksheet, and we are going to look at specific answers. Our first question: name the parts labeled A, B, C, and D. A will be a deoxyribose. Deoxyribose is A. B, very important, B will be a phosphate group. B, a phosphate group. Look, it is not a phosphate. It's a phosphate plus other elements. And then C. We will look at C, and C will be a nitrogenous base. C will be a nitrogenous base, and I will guess that it is either adenine or thymine, because adenine and thymine are bonded by 2 hydrogen bonds. And then D, the bond will be a hydrogen bond that we will have between those 2 bases, and those 2 bases will be one cytosine, the other one adenine. It will be difficult to decide which one is which. We can remember that adenine and guanine are the purines, so they were the larger ones. That is something that we cannot see on this diagram. And then cytosine and thymine are called the pyrimidines, yes, and they were smaller. So if we were able to see a double ring and a single ring, then we could have named it a bit more specific. Okay, so now the next question was, on the diagram, draw a circle. Oh, always take care for this type of questions, where that is in between other, and it is now, there we have a 4 marker. So there we have our circle. Make sure on this side, this will be attached to that one. And there we have our nucleotide. And to get our answers, we need to know that this is the deoxyribose sugar. This will be the phosphate group. And this one, the nitrogenous base. Again, it will be either adenine or thymine due to the 2 bond. If we have circled that one, we would have, say, the nitrogenous the nitrogenous base is either guanine or cytosine due to the 3 hydrogen bonds there. Name the type of chemical reaction that is involved in the formation of a molecule of DNA. A chemical reaction, we have condensation reactions, and we have hydrolysis reaction. So this is a condensation because it is forming a molecule. It's building a molecule. The term used to describe the shape of DNA, double helix for 1 mark. We need to write both. The sequence of the bases in DNA carries information about the order of amino acids to make a particular protein. This is called the genetic code. What is the name for a section of DNA codes for 1 amino acid? It's a triplet. The section of DNA which codes for 1 protein or polypeptide is a gene. Our next question. If the DNA sequence TGA will be threonine, CAT will be valine, GTC histidine. Okay. Right. Oh, and then we have GGG, and that will be proline. That is the DNA sequence. If an error is made when the DNA copy itself, the genetic code will be altered. And now we have a gene mutation. And please, class, we need to emphasize gene mutation, because later we are also going to study a chromosome mutation. So we have to specify here if there is a mistake with the DNA copying, an error, it's a gene mutation. The error results in a change sequence of a base as follows. The TGA, CAT, GTC, GGG is now TGA, still correct. CTT, CTT is now wrong. GTC, GGG. So CTT is now glutamic acid. TGA is still correct. CAT is correct. CAT is wrong. GTC, GGG. Right. And then we look at the definition for transcription, the synthesis of mRNA, from a DNA template by complementary matching of the nitrogenous bases in DNA. Please study this one. Translation, the process of converting the information carried by the mRNA to the correct sequence of amino acids, or the process of converting the information carried by mRNA to form a particular protein. The diagram below shows a sequence of a strand of DNA, which codes for part of a protein molecule. Write down the mRNA. So it's CAA, U, because of the A bonds to U, and this must be in this correct sequence. Now, and TGG will be ACC. Remember that the mRNA codon will always be the opposite to the DNA, the complement. Also remember that thymine only in DNA. In RNA, thymine is replaced by uracil. Right, now the next answer is the anticodon is the opposite to the mRNA and therefore the same that was written on the DNA. And this that I put in brackets is extra notes and will not be part of your question. I just wanted to lead you to it. So, in this question shows the anticodon of 9 different tRNAs. Select and write down from the above the amino acid that would be required for the base sequence of mRNA. So on the mRNA I have U. What will I have on the tRNA? A. You agree? Phenylalanine. On the mRNA I have GUU. So on the tRNA I will have CAA. And that will be valine. AUG on the mRNA. So the tRNA will be UAC. UAC, and there it is. And there we got the answers. So please check where you had a wrong answer, where something was really confusing. Either contact me with WhatsApp or a phone call, or you can even send me an email. Or you can wait until we have a Zoom lesson, which I hope will happen this week. Thank you, grade 12s, and I hope that you have studied well this part of our genetics work. Next time, we are going to discuss the genes and what is happening between having brown eyes or blue eyes if my mother and father has brown eyes. Can that happen? I'm looking forward to research and discover this knowledge with you on our lesson. Stay safe. Bye-bye.
Wash away the germs. Akili and Happy Hippo have come to play with Little Lion. Little Lion doesn't want to play. He's afraid of germs. Germs can make you sick. Don't worry, Little Lion. We will get rid of the bad germs and then you can play with us. They hunt for germs high and low, inside and outside. But the germs are nowhere to be found. What are you looking for? Asks Miss Shrub. We're looking for germs, says Akili. Germs? Germs are everywhere. They are so tiny that you can't see them with your eyes. When you touch something dirty, germs hide on your hands. With water and soap, you can get rid of the germs and make your hands clean. You must wash both sides of your hands, under your nails, and between your fingers. We will wash away the germs and make your hands clean. Akili, get fresh water. Happy Hippo, get soap. Wash, wash, wash with water. Scrub, scrub, scrub with soap. Rinse, rinse, rinse with water, and the germs will be gone. Akili and Happy Hippo run to tell Little Lion that they found a way to get rid of the germs. After we go to the toilet, after we sneeze, when we cook, and before we eat, we wash, wash, wash with water. We scrub, scrub, scrub with soap. We rinse, rinse, rinse with water. And the germs will be gone. Now we can play all day because we know how to keep the germs away. After all that playing, it's time for cake and tea. But remember to always wash your hands before you eat. The end.
In a corner of southern Africa lies a vast and sparse land, where endless horizons, dramatic scenery, and untouched wilderness abound. (1,500 kilometers of harsh desert coastline, a mosaic of biodiversity, Sub-Saharan savanna, acacia bushland, encased by 2 deserts. Home to wildlife and wilderness abundant, yet ever at risk. This is Namibia, home to some of the most noteworthy conservation success stories across the globe. Initiatives forged, manned, and driven by passionate individuals and groups.
People are busy there, walking for more than 45 kilometers a day.
Special species and wild locations under constant threat from an increasingly destructive developing world.
One of the world's true survivors.
This is the story of the Namibian wilderness, the stories of the enigmatic wildlife and natural wonders of her realm.
They are not a threat to anyone, if you understand them.
This is the story of those determined to preserve it, and thereby themselves.
Don't think I want to tell my children one day that we had rhinos in Namibia. I want to show them live rhinos.
This is the story of the last defenders.
We have been tracking them for about 7 days, and on the 7th day, we come across the butchers, and there was a shooting. We are not armed. We having a camera and GPS, we are just hiding behind the bushes. If I don't protect this species, who's going to do that? So is that why I sacrificed my life to go on conserve the rhinos.
Armed out in the field with basically a notebook against poachers that are armed with highly dangerous weapons and more. How is this an equal fight?
I started right on the ground as a tracker, and I worked all the way up to be the CEO now, and I know how hard it is. It's commitment and dedication and the patience that you have for the animal. It's really hard. It's like you had something in your house that you are protecting for so long, and when you go away from home, somebody comes and kills you.
These Dishandi van die cherries in it. I want to say thank you to the Indica Vibe for having the nation's favorite Nama boy. Soms die zal ik from a fun distance af. Me being in this game, some sinds die goede gebeur and soms is the biggie on a lay low.
I think it's fair to say that you should continue pushing this SMI for
News just in. Brace yourselves as One Africa TV's Today on One, our local current affairs lifestyle program, brings you a newly revamped look and feel for the nation at large.
You talk, we listen.
The way they operate have to be different because we are losing lives here.
With over 1 million impressions on posts, we bring you
Community Voices,
Sport, Consumer Watch,
social media commentary,
the weather, and so much more.
Today on One, always there.
Enaranje wa midenzelo kudako Langarans Pharmacy. Atuyo shajima tushua turimo mburuma yokutira omushithe mara uruvede ndwimaru ya arukapita oushanga tukonjithe okukara polonganda atutira aturuma ngatukonjithe okukara polonganda shitwa piti rumwe matuka eneno kuyeto mushithe uyeruke mondondo oushanga tukonjithe okukara polonganda turitire modonju arikane aturoro kuru mburuma maitupo inavya ngatukare polonganda rikane.
Mama!